Our next presenter is Dr. Ruth O'Regan. She's going to share her talk, Precision Treatment of Breast Cancer. Dr. O'Regan, Dr. O'Regan is the chair of the Department of Medicine and holds the Charles A. Dewey Professorship at the University of Rochester. Dr. O'Regan also serves as a professor of oncology, physician-in-chief at Strong Memorial Hospital, and associate director for education and mentoring at the Wilmot Cancer Institute. An internationally recognized breast cancer specialist and academic leader, Dr. O'Regan previously served as division chief of hematology, medical oncology, and palliative care at the University of Wisconsin School of Medicine and Public Health. Across her roles, she has been deeply committed to mentoring the next generation of physician scientists and advancing cancer care through research and education. Welcome, Dr. O'Regan.
Not mine, anyway. Thank you very much, Sarah, and thank you all for being here to celebrate 100 years of the medical and nursing school. So I'm going to kind of follow on from what Steve talked about and focus a little bit more specifically on breast cancer and specifically metastatic breast cancer, and try and, which following what Steve as well just said, the importance of clinical trials and drug development. Just a few stats to start off with. So the current statistics are that 1 in 7 to 1 in 8 women up to age 90 will be diagnosed with breast cancer through their lifetime. The outcomes have improved through greater awareness. That's really through the breast advocacy group, early detection through screening, and more effective therapies. However, unfortunately, we still lose more than 40,000 patients a year because of metastatic disease or disease that's spread outside the breast. I know this is very simplistic, so I apologize for this, but just in terms of why cells become cancerous. So we know that normally cells divide into 2, into 4, etc., and become healthy tissue. In cancer, what happens is the normal cell develops some kind of genetic change that results in cancerous cell division, which then results in uncontrolled growth and a malignant tumor. So what we're going to talk a little bit about, which Steve already mentioned, is what happens in these genetic changes and how can we therapeutically target those changes to improve outcome for patients with cancer. So really our goal in oncology, and I would say this is across all of medicine actually, is to come up with personalized or precision therapy. So what you want to do is find out what's special about the cancer cell, what's a target in the cancer cell or a gene that's gone awry that we can then develop a therapy towards. And the important thing is that whatever that target is, it shouldn't be on normal tissue, because what you want to do is have the most effective treatment, but avoid unnecessary toxicity due to off-target effects. Now, I will say, and I'll give you an example of this in a few minutes, we have historically had a one-size-fits-all approach in treating patients with cancer. What I mean by that is when a drug comes out, we give it to every type of cancer. We really weren't very smart about things, but we have over the last couple of decades moved much more towards really looking for targets and focus on these targets. So looking at the bullseye, we want to be outstanding in the middle where it's a perfect drug. We're probably good or very good at this time point, so we still have quite a bit of work to do. Now, just for complete, here's an example of what is not precision medicine, and that is chemotherapy. And unfortunately, chemotherapy still remains the mainstay of a lot of the treatments that we give. So I think all of you probably know how chemotherapy works. It just looks for cells that are dividing rapidly, which is a hallmark of cancer cells. Well, that's fine, but the problem is there's lots of cells in the body that divide rapidly, including our hair. That's why patients lose their hair from some chemotherapy drugs. Notably, we do have cold cap machines in a number of our sites, which are effective with some chemotherapy agents. On the GI tract, that's why patients get nausea and diarrhea. And then for us, what's sometimes the most concerning is the impact on the bone marrow. So about a week after you get chemotherapy, your white blood counts drop, and that means that patients are unable to fight infections like they would before. On top of that, there's a lot of specific issues and more chronic issues that can happen with chemotherapy, including damage to the heart and neuropathies, for example. So we are trying to move gradually away from chemotherapy, but it's still something that we use very often. Now, I would say that breast cancer has really been a trailblazer in the idea of targeted therapy. So the whole history of the estrogen receptor is actually very interesting because the first association between the ovaries and the breasts was actually discovered in Scotland by a farmer back at the end of the 1900s. And what he found was that if he took out the cow's ovaries, they stopped making milk. And then a surgeon in the UK basically thought this might work for women. So he had some premenopausal patients that came in with breast cancer. He took their ovaries out and their cancers regressed. Well, at that time, nobody knew why that was happening. But you can see about 50 years later, the estrogen receptor was actually discovered in the rat uterus and then was cloned about a year later. So about 2/3 of breast cancers express estrogen receptor. And there was a number of years before the idea that these estrogen receptor positive breast cancers were driven by estrogen before we actually noted that particular finding. So here's an estrogen receptor shown here. What estrogen does is it binds the receptor, activates the receptor, and results in cancer cell proliferation. So what did we do to turn that off? We had a number of agents that we used prior to the tamoxifen coming along. Tamoxifen came along, in the 1970s or so. And what tamoxifen does is it binds the estrogen receptor, but it doesn't activate the gene. So it stops the cancer cells actually proliferating, and really has been an incredibly effective drug. And one of the first certainly the first targeted agent for breast cancer, but one of the first targeted agents for any cancers. It was approved back in 1977. So we've had it for nearly 50 years for metastatic breast cancer. Subsequently, it was approved to treat early stage breast cancer to prevent recurrence, and also is used to treat women at high risk to prevent breast cancer. So that's one, and I will say that we have multiple agents that through the last several decades that target the estrogen receptor, so it's still a very viable target. But this really is our success story in breast cancer, the HER2 alteration. So the HER2 receptor was discovered back in the 1980s. It's overexpressed in about 1/3 of breast cancers. And what you can see here is this is a HER2 positive breast cancer. It's got lots and lots of HER2 on the surface. The problem with this is that it's associated with a very aggressive form of breast cancer that has a very high propensity to come back as metastatic disease within 2 to 5 years of diagnosis, and also has a very high propensity of going into the brain. So about 1/3 of patients with HER2 positive breast cancer historically have developed brain metastasis. That's our nightmare as a medical oncologist because it's very, very difficult to treat. The good news is right after HER2 was discovered, this antibody was developed. It's a humanized anti-HER2 antibody, which many of you have probably heard of, called trastuzumab or Herceptin. The HER is obviously because of the protein. So the antibody targets the HER2 protein with very high affinity and specificity. It's very, very well tolerated. It really has no toxicity because what you're seeing in the gray there is it's 95% human, so there's very low risk of immune reactions. It has some activity on its own, but when you give it with chemotherapy in the lab to HER2 positive cancer cells, what happens is they're completely eradicated. It's really an incredibly effective treatment. So obviously the next step was to look at this agent in clinical trials and to do it with chemotherapy. This clinical trial basically looked at trastuzumab or Herceptin in addition to chemotherapy in patients with metastatic HER2 positive breast cancer. There's an interesting story about this because remember I said in the beginning that at this particular time, any new drug that came out, we gave it for every type of cancer. There was nothing specific. But Dennis Lehman, who had discovered the HER2 receptor and was developing trastuzumab, went to the FDA with the breast cancer advocates and really fought for this drug only being used in patients with metastatic HER2 positive breast cancer or breast cancer overexpresses HER2. And eventually they did listen to him. But it actually took quite a long time actually to get the trial off the ground. And thank goodness they did, because this is the results of the trial shown here. And it was basically patients with HER2 positive metastatic breast cancer who were treated with chemotherapy alone or chemotherapy plus Herceptin. This curve is looking at how long their metastatic disease was controlled for. And you can see here the curve split quite quickly in favor of the patients who got Herceptin with chemo compared to chemotherapy alone, for a difference about 5 months or so, which may not seem that much. But the important thing here is that in the control arm, the breast cancer advocates were very, very vocal, and the patients in the control arm, many of them went on to get Herceptin afterwards. So you can see here at 40 months, 3/4 of these patients had got Herceptin. So that would certainly have muddied the survival results from this trial. So you may wonder, well, what would have happened if he hadn't been so dogged about insisting that this drug be looked at only in patients with HER2 positive cancers? Well, it wouldn't have worked, because this was a study that I think rather stupidly we did after this for patients with HER2 negative breast cancers. Because remember, Dr. Slayman said this only works in HER2 positive breast cancer. Identical design. The only difference here is the patient's cancers did not have overexpression of HER2. This again is looking at disease control. You don't see that split of the curves at all. There was no significant difference. So we would have thrown this drug out and we wouldn't have it today. Subsequently, of course, the next move was to look at it in patients with early stage HER2 positive breast cancer. And this is really where it made its mark. So here is looking at recurrence rate shown here. And what you can see here is the patients who got chemotherapy with Herceptin had a 12% lower recurrence rate than patients who got chemotherapy. So you can see out of 5 years, actually this is nearly 10 years, you can see only about a quarter of these patients have had a recurrence compared to 40% in the chemotherapy alone arm. It also impacted survival, as you can see shown here. The difference is about 9%, so 84% survival in the patients who got chemotherapy plus Herceptin compared to 75% of the patients who did not. So these patients are actually being cured from this cancer because of this drug. So if anybody's interested, this whole story of Herceptin was captured in a Lifetime movie called Living Proof. Harry Connick Jr. played Dennis Slayman. So I guess if you don't win a Nobel Prize, which he actually didn't in actual fact, this may be the next best thing. So if you're it actually is kind of an interesting movie because it really talks about the breast cancer advocates because they were so pivotal in getting this drug to patients. Now, since that time, we have loads of agents that target HER2. Other antibodies, I'll briefly talk about that, small molecules. We also have these very newer drugs called antibody drug conjugates that are going to replace chemotherapy. What they are is they are antibodies that target something on the cancer cell and have chemotherapy attached to them. So the chemotherapy gets deposited right into the cancer cell. They're not quite there yet, but eventually I think they will have a very favorable toxicity profile. And I just to show you where we are in terms of survival. And these are patients with metastatic HER2 positive breast cancer, so incurable metastatic breast cancer. This is another trial where the standard arm now was Herceptin plus chemotherapy because I showed you that was so effective. The study arm was trastuzumab chemotherapy and a second antibody called Perjeta that does not increase toxicity in any way. And what you can see here, this is looking at survival, not disease control. You can see here that by adding in this non-toxic second antibody, there's a difference of 16 months in how long these patients are surviving. But perhaps more importantly, when you look at the patients who got the 3 drugs, you can see here that their median survival is almost 5 years. And now, obviously, there's some patients that would die earlier than that, but there are also patients that we have in our practice that live with this metastatic breast cancer for decades. In fact, the question that comes up is, we have to continue them on treatment because they've been on it so long. So this is really a huge success rate with these agents. And again, as I mentioned, and I should mention as well, the chemotherapy is only given for about 6 months. Then the patients end up on maintenance antibody treatment and have really pretty much normal lives. So this is kind of where we were in 1998. And then with the sequencing of the whole genome, it kind of came the genomic era, which allowed us to really take a much deeper dive into kind of genes and proteins that were driving cancers. And because of all this work, this is kind of a slide showing all the agents we have to treat breast cancer. We have new chemotherapy agents. We have immune therapy, which is where Steve mentioned this, where you're trying to turn the patient's immune system on the cancer cells. And this is Ryan's specialty. But you can see in the bottom of this slide, these are all targeted agents. So these agents were all targeted towards something in the cancer cell that's been determined by sequencing. So with all these agents, we're done, right? But unfortunately not, because as I told you in the very beginning, despite all these new treatments, earlier diagnosis, all these things, we're still losing 40,000 patients annually to breast cancer. So there is more work to be done here. And the reason, what happens with all these great drugs that we have in the metastatic setting, and sometimes in the early stage setting, is that they stop working. And they stop working because cancers are very smart. And they basically outsmart the drug that they're being given. So this is an example of an estrogen receptor positive breast cancer. You can see the estrogen receptor here in the nucleus shown here. So like if you take tamoxifen, for example, you give it to a patient with metastatic disease, eventually it's going to stop working. The 2 main reasons it stops working or develops resistance is firstly loss of target proteins. So you could lose the estrogen receptor. It doesn't happen very often, but that does happen in cancers. but more often you get mutations of the receptor. So mutation of the estrogen receptor are well described, and that means that endocrine therapy like tamoxifen doesn't work anymore. The other thing is that all these other proteins in cells, some of them take over and start driving the cell. And I'm going to mention this one here, PI3 kinase, because what we've shown is that if you target this pathway here, so if you basically block these different proteins, what happens then is the estrogen receptor is actually restored, and it actually gets, it's resensitized to endocrine therapy. So it's kind of akin to these super bacteria that I think we've all heard about, where, you know, as bacteria get exposed to antibiotics, they develop resistance to these antibiotics, and you end up with this horror here, this methicillin-resistant staphylococcus shown here. So not only can they make patients very sick, but the antibiotics that we need to use to treat these bacteria are incredibly expensive. So just to talk about clinical trials briefly, because really, we talk about all these therapies, how do we get them to patients? They all go through very rigorous studies through clinical trials. The early trials make sure the drugs are safe and work out what's the correct dose. Then we look to see if they're effective in patients with specific cancers, but then we have to confirm that they're better than the standard of care, so the drugs that we were using previously. Very important take home, which probably most of you know, is that you do not see placebo or sugar pill, but placebo only arms in cancer clinical trials. This means that patients at the very minimum are going to get standard treatment. So there's nobody getting no treatment if they enter a cancer clinical trial. So this is kind of the journey of new drugs to patients. So discovery, laboratory evaluation, I put 3 to 5 years. We all know it's way longer than that, right? So then they go to phase 1, which is where you look at safety. Now, even though these are small trials, they take a while because you have to see how patients do at each dose level. Then moving on to phase 2, a little bit larger, looking to see if they're effective in a particular cancer. And then phase 3 are much larger trials where you're comparing to standard of care. So you can see this takes a long time, at least 10 to 20 years. And of course, it's really expensive. One of the reasons it's very expensive is because a lot of the drugs will fail at phase 1 or phase 2. So this is one of the reasons that the new drugs that we have tend to be so expensive. So again, just to give you an example, I mentioned already about let's go back here, about the genetic changes. So with the technology we have right now, you can really do a very deep dive on cancer. So these are metastatic ER-positive breast cancers compared to the primary breast cancer. And you can see here that there are lots of alterations here, some of which we have therapies for. Just to mention a couple, here's ESR1, that's the estrogen receptor. I mentioned that mutations of the estrogen receptor can occur. There are now drugs that target that. And then down here, we have ERB2, which is HER2, and ER-positive breast cancers. Some of them actually develop mutations in the HER2 receptor as they're treated with endocrine therapy. And the good news with that is that all those agents I showed you earlier can be used in that setting. But I'm going to focus on this PIK3CA mutation or PI3 kinase mutation, because that's present in about 40% of estrogen receptor positive breast cancers, both primary and secondary. So we spent many years trying to get a good drug that would target these PI3 kinase mutations. Eventually, we ended up with this drug, alpelisib. So what's happening in this clinical trial is these are patients with metastatic breast cancer. They had to have tissue available for analysis to see whether they had a mutation of the PI3 kinase or not. That doesn't make any sense, I know, but that's what was done. And then they got randomized to endocrine therapy alone or endocrine therapy with the PI3 kinase inhibitor. So guess what happened here? Well, of course, as you would expect, the patients who did not have the mutations, the disease control is no better when you give this PI3 kinase inhibitor. However, if they have the mutation, you can see here the curves separate. And there's about a doubling in disease control for patients who get the PI3 kinase inhibitor compared to those did not. So very similar to the Herceptin story I told you earlier. You'd think we would have learned our lessons, but obviously we continue to make mistakes. So as I said, I think that basically these are all the agents for ER-positive metastatic breast cancer. I think all the future agents will come out of tumor genetics, or I think immunology is the other big area. So I'm sure walking in here, nobody saw the sign that we have designation from the National Cancer Institute. It's all over the place, right? But it's very exciting. This is just a photograph of us after the site visit when the NCI visitors came. What does it mean? I think, as probably most of you know, it's the highest honor a cancer centre can receive and puts Wilmot in the top 4% of cancer centres nationally. And here's Jonathan at the time of the announcement. But very important in our designation are our clinical trials. So we have many clinical trials available across all different cancer types. There's the web link. Also, if you just Google Wilmot clinical trials, you can find them. So the one thing I would say is, it doesn't, whether you loved one has cancer, a chronic disease, or any nature, do ask your doctor about a clinical trial, because as Steve said, it can help you or your loved one, but also what we learn from clinical trials is just so fundamental because of all the agents that I showed you today. So I think we can be very positive here, because with all the technology we have right now, rapid discovery of new agents through genomic analysis is going on, and that will result in continued improvement. Continued improvement for patients with metastatic breast cancer and other cancers. And I think it's inevitable. So thank you for your attention. They're a very quiet bunch. I think they want their lunch. Hey, Tanya.
Hi, Ruth. I'm actually thinking about, the cross-fertilization that can happen at URMC between what Steve said and what you just showed on your slide. And although the dark genome and the retrotransposons are bad in aging, et cetera, can contribute to these diseases. In breast cancer, if TP53 is one of your top mutations, that actually most of these retrotransposons are repressed in expression. But with the TP53, it gets re-expressed, very specifically only in cancer. And I believe in breast cancer and, for example, ovarian cancer, the line 1 element, ORF1p, is very highly expressed. So now we can think of it as a target for immunotherapy. And that's something that, we can look at different cross-fertilization of different fields that URMC has real power and expertise in. And we could make some very novel discoveries.
Thank you for that comment, because as you know, P53 is something that we just haven't really been able to target or use to our advantage in treating patients. So I think that'd be very interesting. Immunotherapy, at least for estrogen receptor positive breast cancers, is pretty ineffective because they're kind of immunologically cold. So there may be this connection there. So very interesting. All right. Thank you very much.